For part 1 of this tutorial, we will explore microarray datasets in the Gene Expression Omnibus, or GEO, and use the GEO to R tool to calculate differentially expressed genes. The first thing we do is open the GEO website and type in the experiment we want to explore. In this case, we will explore experiment GSE 76278. Type it into the search window and search. The summary and overall design give you a quick idea what the study is about. In this study, the transcriptomes of plants with a mutation in the WRKY1 transcription factor and wild-type plants are compared under combinations of light and nitrogen treatments. We can also learn what experimental platform was used to process the samples. In this example, it was the Affymetrix Arabidopsis ATH1 genome array. If you expand the samples, you can see which experiment corresponds to which microarray chip. To further explore this dataset, we can open the series matrix file, which is a gzipped file, so you will need a program to open it up. Save this file in your desired folder. Then use a program like 7-zip to extract the file. Once extracted, you can then open the series matrix file using a program like Excel. Here we can find additional information about the experiment, for example, which normalization method was used, as well as specifics about the experimental conditions. If we expand column B and look at row 56, we can see that the GCRMA package and R was used to normalize the expression data. If we scroll further down, we can then see all of the normalized expression values. Now let's go back to GEO to perform differential expression analysis using GEO to R. Click Analyze with GEO to R. All of the samples will automatically be loaded, but for this tutorial we only want to compare wild-type and mutant plants under control and nitrogen treatment conditions. We do this by first defining the groups we are interested in analyzing. So click Define Groups and type wild-type control and hit Enter. Mutant control, wild-type nitrogen, and mutant nitrogen. Now we can assign these groups to the samples we want to analyze, which are the samples GSM 1978990 through GSM 1979001. So we'll select GSM 1978990 through 992 and assign those to wild-type nitrogen. Next, we'll select GSM 1978993 through 5 and choose mutant nitrogen. The next three we'll select and assign those to wild-type control, and the next three we'll select those and assign them to mutant control. Click on the Options tab at the bottom to see which test for multiple correction is selected. We will use the default for false discovery rate the Benjamini and Hochberg test. Now we can click on value distribution to see that the samples were normalized properly prior to differential expression analysis. Look for the median of the box plots. You want those to be at about the same place to see if the samples had been normalized properly. Now, on the Geo to R tab, click top 250 to run the analysis. Geo to R uses Lima to perform differential expression analysis. The results are organized from most significant adjusted p-value to least significant. Geo to R also converts Affymetrix probe IDs into gene symbols and provides their description as well. Let's go ahead and save all of the results. Click Save All Results, and you'll want to save this as a text or CSV file. If you're using a PC, the simplest thing to do is to paste all of the results into a text editor like Notepad and save it as a text file. We now need to apply a cutoff to obtain a set of differentially expressed genes for downstream analysis. The probe IDs that fall within the chosen cutoff value are those that you have confidence in their differential expression. For this reason, it is best practice to use the FDR-adjusted p-value to determine your cutoff value, and this takes into account multiple comparisons. So for example, an adjusted p-value cutoff of 0.01 means that you will reject the null hypothesis 1% of the time just due to random noise in the data. To get started, let's open the saved DEG results in Excel. Let's scroll down the adjusted p-value column to see how many genes are differentially expressed with an adjusted p-value less than 0.01. A cutoff of less than 0.01 gives us 861 genes to work with. We want to select only the probe IDs that meet this level of significance by selecting cells 2 through 862 and copy them into a new worksheet that we're going to call SIG IDs. Now, let's open the series matrix file that we saved earlier. We want to select only columns of data that we're interested in and then copy and paste them into a new worksheet called Trimmed. Scroll down until you see the sample names, and we're only interested in samples GSM 1978990 through GSM 1979001. Copy and paste those into Trimmed. Now let's create a file with expression values for our significantly different genes. We're going to do this using the VLOOKUP function in Excel. The first thing we need to do is copy the sample names from the series matrix file and then paste this into the top column of SIG IDs. Now in cell B2, let's type equals VLOOKUP, open parentheses, and select the first probe ID in column A, and then type a comma. We then need to select the trimmed data sheet and type a comma. Now type the number 2, which designates which column in trimmed you want the formula to search. Next, type a comma and the number 0 to indicate that you want an exact match for the probe ID of interest, and then close the parentheses and hit enter. We have to make one final adjustment to this formula. In front of the value A2, type a $sign. And so now the equation will always reference this cell, and hit enter again. You can then copy and paste this formula for each sample, but we need to change the column number that we want the formula to look for in the trimmed data sheet. So now for sample 2, we want the formula to search column 3, and for the third sample, we want it to search column 4, and so on. Next, we can copy the equations for each sample into all of the rows. To be able to use this file for downstream applications, we need to copy this sheet and paste it into a new workbook that we're going to call significant values, because we want to save only the values and not the equations. Now, just save this new sheet as sig values as a separate tab delimited file, and then you're ready to use it for downstream applications.